Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before starting the topic, please like, share, and subscribe the channel. In the last session, we studied about structural and functional relationship. In today's session, we will study isoelectric pH, precipitation reactions of proteins, and denaturation. Significance of isoelectric pH. The amino acid composition will determine the isoelectric pH, that is pI, of protein. The alpha amino group or carboxyl group are utilized for peptide bond formation and hence are not ionizable. All other ionizable groups present in the protein will influence the pH pI of the protein. At the isoelectric point, the number of anions and cations present on the protein molecule will be equal and the net charge will be 0. At the pi value, the proteins will not migrate to any electric field. At pi, the solubility, buffering capacity, viscosity will be minimum and precipitation will be maximum. On the acidic side of the pi, the proteins are cations, and on the alkaline side, they are anions in nature. The pi of pepsin is 1.1, casein is 4.6, human albumin is 4.7, human insulin is 5.4, human globulins is 6.4, human hemoglobin is 6.7, myoglobin is 7, and lysozyme is 11. In this diagram, we can see that on adding acid and base to the amino acids, they become cations and anions respectively. Acidic dyes such as eosin will dissociate H plus dy minus, which will then attached to protein NH3 plus, that is protein cations, whereas basic dyes such as hematoxylin and methylene blue are dissociated to OH minus plus Di plus, which will then stain protein COO minus, that is protein anions. The staining characteristic of a protein is determined by the pi of that protein. In this diagram, we can see that on adding eotoxin, eosin protein becomes an cation, whereas on adding hematoxylin, protein becomes an anion. Chemical synthesis of peptides. Peptides are artificially synthesized for the following purposes. Number 1, to check whether the sequence analysis is correct or not. Number 2, the primary structure of a protein is altered by one or two amino acids so as to determine the biologically important area or the active center. Number 3, to get pure preparations for medical and diagnostic purpose. For example, HIV antibody in the blood of AIDS patient is detected by ELISA method. For this, pure antigen from HIV is to be coated in the test tube. Preparation of quantity of antigen from the virus is tedious and hazardous. The best way is to synthesize the antigenic part of the protein. In this diagram, we can see that in ELISA, HIV ELISA test result, A indicates uninoculated column, B indicates negative control, whereas C indicates positive control. Commercially, it is cheaper to synthesize small peptides than isolating them from biological sources. It, it is simplified the proteins to process by adding carboxy terminal and amino acid to insoluble polystyrene beads so that washing and purification becomes rapid. In the principle, the carboxyl group of the last amino acid is fixed on the resin and other amino acids are sequentially added. Physical properties of proteins. Protein solutions exhibit colloidal properties and therefore scatter light and exert osmotic pressure. Osmotic pressure exerts the plasma proteins in RT is clinically important. Molecular weight of some of the proteins are insulin 5700, hemoglobin 68,000, albumin 69,000, immunoglobulins 1,50,000, whereas rabbit papilloma virus protein it is 4 crore 70 lakhs. In this diagram, we can see that there is effect of colloidal osmotic pressure amidst the barrier leading to increased volume of colloidal side. Shape of the protein also vary. Thus, insulin is globular or albumin is oval in shape while fibrinogen molecule is elongated. Bigger and elongated molecules will increase the viscosity of solution. Since proteins are made up of amino acids, the pI of all the constituent amino acids will influence the pI of the protein. Precipitation reactions of proteins. Purification of enzymes and other proteins usually start with precipitating them from solution. This stability of protein in solution will depend mainly on the charge and hydration. Polar groups of the proteins like NH2, COOH, OH groups tend to attract water molecules around them to produce a shell of hydration. Any factor which neutralizes the charge or removes the water of hydration will therefore cause precipitation of proteins. The following procedures are used for protein precipitation. First, sorting out. When a neutral salt such as ammonium sulfate or sodium sulfate is added to the protein solution, the shell of hydration is removed and the protein is precipitated. This is called salting out. As a general rule, the higher the molecular weight of a protein, the salt required for precipitation is lesser. In this diagram, we can see that there is salting in and salting out in the graph. Thus, the globulins are precipitated with half saturation of ammonium sulfate, but albumin will need full saturation with ammonium sulfate for complete precipitation. Second is isoelectric precipitation. Proteins are selected least soluble at their isoelectric pH. Some proteins are precipitated mainly when they are adjusted to their isoelectric pH. The best example is casein from a flocculent precipitate at pH 4.6 and is redissolved in high acidic or alkaline solutions. In this diagram, we can see the machinery of isoelectric pH precipitation. When the milk is curdled, the casein forms a white curd, while lactic acid produced by the fermentation process lowers the pH, the isoelectric point of casein. Precipitation by organic solvents. When an organic solvent is added to the protein solution, water molecules available for proteins are reduced and precipitation occurs. Organic solvents reduce the dielectric constant of the medium, which also favors the protein precipitation. Hence, alcohol is a powerful protein precipitating agent. This may explain the disinfectant effect of alcohol. Fourth is precipitation by heavy metal ions. In alkaline medium, proteins have net negative charge or they are anions. To the to such a solution, if the salts of heavy metals are added, positively charged metal ions can complex with protein molecules and metal proteinates are precipitated. Salts of copper, zinc, lead, cadmium, and mercury are toxic because they tend to precipitate normal proteins of the gastrointestinal wall. In this diagram, we can see that by adding heavy metal ions, the proteins are precipitated. Based on the principle, raw egg is sometimes used as an antidote for mercury poisoning. Next is precipitation by alkaloidal agents. Tungstic acid, phosphotungstic acid, trichloroacetic acid, picric acid, sulfosalicylic acid, and tannic acid are more powerful protein precipitating agents. These acids lower the pH of medium when proteins carry net positive charge. These protein cations are complexed with negatively charged ions to form protein tungstate, protein picrate, etc., and thick flocculent precipitate is formed. In the clinical laboratory, phosphotungstic acid or trichloroacetic acid are usually used for precipitating proteins. Tannin in leather processing is based on the precipitating effect of tannic acid. Under certain conditions, proteins undergo denaturation, which is a mild form of precipitation reaction. Heat coagulation is an irreversible precipitation process. In this diagram, we can see that tannic acid is used to precipitate the protein. Denaturation of proteins. Mild heating, treating with urea, salicylate, X-ray, ultraviolet rays, high pressure, vigorous shaking, and similar physico-chemical agents produce denaturation. There will be non-specific alterations in secondary tertiary, quaternary structures of protein molecules. Primary structure is not altered during denaturation. In general, during the process of the solubility process, the is decreased while the precipitability of the protein is increased. It often causes loss of biological activity. Native proteins are often resistant to proteolytic enzymes, but denatured proteins will have more exposed sites for enzyme reaction. Since cooking leads to denaturation of proteins, cooked foods are more easily digested. Denatured proteins are sometimes renatured when the physical agent is removed. Ribonuclease is a good example for such reversible denaturation. In this diagram, we can see a denaturation and renaturation of proteins. Immunoglobulins are dissociated when treated with urea. When the urea is removed by dialysis, the subunits are reassociated and biological activity of immunoglobulins is regained. But many proteins undergo irreversible denaturation. For example, albumin, when heated, cannot be renatured by cooling. Here ends our session. Kindly like, share and subscribe the channel. Thank you.